Welcome to ASM Live, the talk show, live from the American Society for Microbiology meeting in Denver, Colorado. We welcome you here to talk about all things microbiological. And today we have a couple of guests. We're going to be talking about the importance of microbes in the environment and one aspect of that focused on microbes and the subsurface environment and how it's influenced by things like fracking. Very important and timely topic today. I'm your host. I'm Stanley Maloy. I'm the Dean of the College of Sciences at San Diego State University and chair of the editorial board of Microbe magazine. And before we begin, I'd like to ask the audience, please feel free to ask any questions. If you have a question, please come up to the mic so that everyone can hear your question. And if you're in the online sphere, please send your questions to hashtag ASM 2013. So let's begin. We have with us today Paula Moser from Ohio State University and Erin Lipp from the University of Georgia. So Erin, could you tell us a little bit about your convening a session that's focused on this important topic of environment and microbes? Tell us a little bit about it.
So the idea is the environment is obviously critically important, and sometimes the microbial component is under-recognized. It has such a fundamental importance in terms of how the environment functions. So the idea of the session is really to look at not only protection of all forms of our environment, whether it's oceans, water, soil, land, the subsurface, the atmosphere, and how that is, protecting of that environment is so critically important to our future going forward, especially in terms of what the microbes are doing. But also on the other side, if we look at the potential for microbes to help us in terms of bioremediation, production of energy, several aspects. And then encompassing all of that is the idea of climate change and how microbes are controlling that or helping to remediate climate change.
This is a pretty broad topic. Hey, what did you do in your personal research? How did you get to this understanding?
It's a very, very broad topic. So my personal trajectory to this point is I actually have a degree in marine science, but I work in public health. So I sort of bring together a lot of worlds as it is. But personally, I'm interested in watershed protection and water quality, especially from a public health perspective and microbial ecology of pathogens. So to me, it sort of fits in the idea of a sustainable environment, but of course, sustainability is much larger than just that piece.
And Paula, tell us a little bit about what you're doing.
Okay, our research group is focused on subsurface microbiology, and we're really interested right now in how different aspects of shale energy development might be influenced by microbial communities in the shallow subsurface and also in the deep subsurface.
And how did you get to this process, and what made you interested in this problem?
Well, all of my degrees are in environmental engineering, so I come to environmental microbiology through graduate training and not as a discipline. And so right now I'm at Ohio State in the Department of Civil, Environmental, and Geodetic Engineering. And because energy extraction is an important part of engineering, and microorganisms play a role in influencing the efficiency of that process, and also in terms of release of some of the different fluids and treatment of the fluids, environmental engineers are really the stewards of how that water is managed.
It's great to hear about engineering departments with people working on microbiology. But you know, I think that it's not obvious to a lot of people that microbes would play a key role in the process of energy recovery from the subsurface. What is the kind of role that microbes play here?
Well, right now it's critically uncharacterized. We really don't know a lot about the role that they're playing, but we know it's really important. For example, if we happen to have accidental releases of these fluids, they're primarily made up of water, but also a small fraction of organic compounds are within the fluids that are used. How biodegradable are those in the environment? What's their fate? And then at depth, these fluids are being injected into the subsurface in large scales. And if those fluids are readily biodegradable, biodegradable, there shouldn't be a big need or a large public concern for the fate of those fluids. However, if they're not biodegradable, then that should be something that we learn a little bit about and possibly alter the process or the chemicals that are used to make it a little bit more on the sustainable side.
So when you say biodegradable, it's not just that they're degradable, but the time frame in which they're degradable, right?
Relative to the transport life of the fluid.
Right,
yes.
absolutely. And so what have you learned so far in your work on this topic?
Well, we're coming at this from two different aspects. We are looking at the degradability of what we're calling a synthetic fracture fluid in the laboratory using typical wastewater bacteria and also typical soil and groundwater bacteria. Wastewater bacteria can readily degrade our synthetic frac fluid. In fact, more than 90% over a very short period of time, less than a week, is mineralized to carbon dioxide. Once we are looking at soil and groundwater communities, the rate at which that's happening is a little bit slower, and redox gradients affect how quickly that's occurring. But again, within about a month period, we're seeing about 80 to 90% of that organic carbon being mineralized. And so just at first glance, it looks like these types of fluids and compounds that are being used don't appear to stick around for that long in the environment, but there is a small fraction that seems to be persistent, and we're going to be looking in more detail at what that is.
The small fraction that's persistent, are they compounds that are of particular concern or not?
They might be. So the characterization of these constituents is not a trivial problem. The EPA is studying this at a very large scale. And so there are many different classes of organic compounds that are used. And so we are trying to basically focus in on the ones that might pose the greatest risk in our degradation studies.
So Paula, the microbes you talked about are the normal microbes in that environment that are taking on this special job, or extra job, if you will, right?
Yeah. Yes.
So you might expect that some different types of environments would be better at degrading the compounds than others. Is that what you found?
So we're building up to be able to look at this across a redox gradient and a salinity gradient and a temperature and pressure gradient. And so I guess what we're expecting is that these compounds might be pretty readily degraded at the surface. But once we have higher salt concentrations, higher pressures, higher temperatures, we might see a slower or a reduced rate of extent of attenuation of these compounds, say at depth, for example.
So in the example you've talked about, a really important role of the microbes is the turnover of these organic compounds. Do microbes play another role in this process?
Maybe you could be more specific.
So, for example, does the microbial composition influence in some way the permeability or the distribution or any other aspect of the process?
Definitely. The microorganisms that are actually being sent down these wells during the hydraulic fracturing process can detrimentally affect the wells in a couple different ways. In fact, one of the compounds that's used in the fracturing fluid is a biocide to try to knock out or reduce the number of viable cells at depth, because if these organisms flourish when they come in contact with some of these hydrocarbons and other labile sources of carbon that are used in these fluids, they can bloom in the subsurface potentially and cause clogging, reduce the permeability, and therefore reduce the amount of hydrocarbons and gas that are going to be coming from these wells. And so if you're talking about a $5 million investment, you want to make sure that that kind of thing doesn't happen. And they can also be involved in starting corrosion processes if they're producing certain types of acids or sulfides in the subsurface. So they can be producing products that detrimentally affect the well in other ways.
Mm-hmm. But the biocide itself could be problematic at a later stage if released into the environment, correct?
It could potentially, I haven't seen much information on that, and maybe Erin can comment a little bit about that.
Yeah, and I don't know too much about the biocides either, but I would think that any of these things could have potential downstream effects that would need to be investigated. I think that's what's maybe surprising about this whole topic is fracking is relatively new, and I think the amount that we don't understand is still quite vast, and so the work that you're doing is really exciting to start getting at that.
Well, I think that most of the general public, we just don't think about microbes when we think about fracking, despite the fact that it's been in the news so much. And so it's not surprising that people are not attacking this at the level that they could. So Erin, maybe you could comment on what you view as being, as the release of compounds like this in general and their impact on watersheds and the environment, and including not only the local environment, but downstream environments.
Right, so I mean, I think, looking at watershed protection, there's so many things that we need to take into account in terms of what's impacting our water. And there's been a lot of focus that has been on sewage impacts, persistent organic compounds. And so there's a fair amount of research that has gone into understanding those things. But again, as you start adding new compounds to this environment, it can have a variety of effects. It can affect public health directly in terms of our exposure to these compounds. But it can also affect the fundamental diversity of the microbial community that may be there, for which we don't really understand what the downstream effects may be. And especially in the age of genomics and metagenomics, I think that's something we're just starting to understand, is really what the microbial diversity is and how it's functioning. Because in a lot of ways, you can sort of think of that microbial community as an organ into itself in these systems, and they're doing a lot of things that are important. And so as you add these compounds, it could be changing their function.
An enormous problem, because now you're talking about watersheds that cover a very large distance, where different parts of the watershed may have different communities, and they may change over time.
Exactly.
Right? So a very important problem. One of the things that was very interesting that you brought up in the beginning was the impact of microbes on climate change and the impacts of climate change on microbes.
Right.
And I think that ties in a very interesting way, because fracking is associated with the recovery of petroleum products, right? And of course, petroleum products have a very critical role in climate change. But climate change and microbes are another thing that people don't really connect usually. Why don't you tell us something about what do microbes have to do with climate change?
Well, microbes have a lot to do with climate change. I mean, microbes produce methane, and methane is a huge global greenhouse gas of tremendous importance. There's a whole field of research that's looking at how to reduce methane production from ruminants, and microbes are critical in that process. And I'm sure there's many other examples, but that's a very obvious one. And then, of course, as climate changes, as you mentioned, it also affects microbes in other ways. So as you see changes in ocean acidification, we could potentially see changes in the types of microbes, cyanobacteria, small eukaryotic autotrophs can be affected by that as well. Temperature, ocean acidification, changes in carbon dioxide and other climate-sensitive gases.
And so ultimately we have ourselves in kind of a big cycle of the potential of changing a lot of things, both at the local level in the groundwater and at the level of global climate change. An interesting thing that I'd like to have us talk about some more is how microbes can help thwart some of those kinds of issues. But first, I'd like to remind the audience that if you have any questions, please come up to the microphone. We welcome you to ask questions. And if you're listening in online, you can send any questions to Twitter, #ASM2013. So one of the things that you just mentioned, Erin, is the impact of climate change, for example, ocean acidification, on the potential microbes that are associated with those areas. So you also said, well, we may be able to engineer microbes so that you have less methane being released from cattle. But what about some kind of more global, people have talked about some global modifications of the ocean to try to influence the climate change itself?
Yeah, so the oceans are typically considered to be a large sink for carbon dioxide. There's so much primary productivity happening in the oceans that it's been seen as a vast resource for being able to take up atmospheric carbon dioxide. And for many, many years now, there's been this idea of looking at spots in the ocean that have high levels of nutrients where you would expect there to be high primary productivity, but there isn't. These are these high nutrient, low chlorophyll areas. And so there's been interest over the years on geoengineering these areas, basically adding adding iron. Iron is highly insoluble in marine waters, but is absolutely required for a lot of these organisms. So studies have been done to basically put bioavailable iron into these areas and try to engineer the ecosystem to take up carbon dioxide. And I think what they have found, for the most part, is that it's a very short-term fix. The ecosystem quickly adjusts, and part of that is because of the heterotrophic community that's also using those iron products as well. And so as the carbon dioxide is taken up, it's quickly respired and released. But the oceans, they are a vast pump, chemical pump and biological pump for taking down carbon dioxide. But the geoengineering fixes in the ocean have not panned out particularly well so far.
I think a lot of people are concerned by experiments like that because we all know in ecology, you tweak one thing and something else happens that you hadn't anticipated.
Right.
Right. And I think that also is an important tie-in to thinking about the fracking issue, the fact that microbes can play this really key role in cleanup, but it may be that eliminating those microbes might be the best way of recovering the maximum amount of product that you want. So, we have a question from the audience right now.
Well, just getting back to the topic the panelists were talking about a moment ago, is it seems there could be a tie-in between the more advanced and rapid methods that are being applied in some of the oceanic research that I've seen at this conference, and then combined with environmental on-land kind of approaches, maybe looking at frack water. Is there any of that being done? You could comment on? How are samples being taken and analyzed now, and are you looking at some more rapid methods?
We are applying some of the omics techniques to some of the samples that we're receiving from oil and gas wells. To be honest, obtaining samples from these environments has been really a challenge, and especially obtaining samples before the well is fracked. To be able to look at the indigenous microbial communities, that's something that hasn't been done. And so applying omics techniques to some of those samples would be really exciting to benchmark some of the communities and then be able to look at how these engineered activities are changing the environment.
Very much, thank you.
This seems like something, Paula, that oil companies would themselves be very interested in supporting this type of research because it's so critical for product recovery. Are oil companies doing research in this area now?
I believe there has been some research by the oil companies, and a lot of that information is not necessarily released to the public. It's used for internal improvements in the processes. There are industry groups that are funding research, especially in the Texas area and in the Pennsylvania area, but they're really interested in specific questions that affect their ability to recover the highest amount. And so some of these questions that we have as scientists and engineers, such as what's the biodiversity of the subsurface environment and how do things change, aren't necessarily directly related to the questions that they want to answer in terms of getting the most energy. I mean, in the long run, it will probably help them in terms of increasing their efficiency, but I don't know that there's the same kind of drive to answer those kinds of questions. At the same time, to be able to obtain these samples in ways that we can determine how much contamination there is. For example, the DOE has done a lot of work on figuring out, using tracers, whether there's been influence of drilling modes or fluids when the samples are collected. And those things are very time-consuming and expensive to do. And so asking oil and gas companies to do that is asking a lot. So.
Who funds your research?
Right now, the work that I'm discussing today is funded by the National Science Foundation and also a state agency, the Ohio Water Development Authority, who's also interested in water quality impacts of hydraulic fracturing activities.
Paula, it's interesting to think, so you're an engineer, approaching a microbiology problem. Do you think you approach the problem in a different way than a microbiologist who is interested in the same question might?
I definitely do. I think that engineers approach things from an application standpoint, and microbiologists often approach it from more of a scientific standpoint. And I think the major improvements and discoveries are made when we work together to learn from both angles.
So are you collaborating with other scientists on this project?
We are. We're working with microbiologists at the Department of Energy National Energy Technology Laboratory, and she's been really critical in a lot of our work. The graduate student who's been working on this is a biology undergraduate student, so she has that background even though she's working in an engineering department. So we are pretty interdisciplinary in our group.
Mm-hmm. And how did you get interested in this particular problem?
Oh, that's a great question. I grew up in Wyoming, and I've been surrounded by energy extraction. My father was a gas engineer, and as having engineering brother and fathers, women often go into engineering disciplines. When I took the position at Ohio State, I was noticing the ramp up of the hydraulic fracturing, and was very interested in how that might play into some of the research that I'd been doing in subsurface environments. So.
No, that's an interesting story. One thing is really clear: we need more women going into engineering, though. That clearly is a place where there really isn't enough representation. So
that's true.
Maybe we need more brothers and fathers who are engineers, right?
Or mothers who are engineers.
So Erin, why don't you tell us about a couple of the other topics that will be covered in the session you're running?
Yeah, so the session, we're excited because it's sort of this new format for ASM where we have 3 invited talks, and then we have young investigators who are selected from abstracts. So we have Dr. Joan Rose is going to be talking about the water virome and her work on basically looking at the smallest microbes that we know of and how that influences sustainability in water environments. Mike Sadowski, who's our Division Q lecturer, is going to be talking about the microbiome in natural systems, especially in the Mississippi River. We have someone talking about the urban microbiome and the built environment, so going from outside to inside. We, of course, have Paula's talk on hydraulic fracturing, or fracking. We have talks on, I'm having to remember all of these now. Yes, we have, yes, so the Deepwater Horizon oil spill, looking at microbial communities in sand environments. We have somebody from the National Ecological Observatory who's talking about basically putting microbiology into these very, very large-scale models of what's happening on a much broader scale.
Why don't you tell our audience what Division Q is?
Okay, so the Division Q is applied and environmental microbiology, the applied group.
So, and that's a broad diversity of topics.
Yes,
is there
very.
something that unifies them? Is there a common approach or some kind of common question that brings it together?
So there is a common thread, but it's probably hard to pick it out. I mean, the common thread is either the idea of environmental protection towards sustainability, or new efforts that are looking at energy and its role in obviously its role in sustainability going forward. So it's a tenuous thread, but hopefully the session will build together. Because I mean, when you look at environmental sustainability, it's a very, very broad topic. And since microbes are interacting on such a remarkable number of levels, we wanted the session to sort of reflect that as well.
It does.
To me, one of the things that I think is very exciting today, that was less true 20 years ago, is how all of our different disciplines influence each other. So what's learned from the human microbiome can have a big impact on how we approach environmental biomes. And so we have to be talking to each other a lot more than, whereas in the past one division would really stick with the people in that division. I think that's particularly exciting. One of the things that you brought up, though, is the Deepwater Horizon oil spill. And it's a very interesting case to think about relative to the subsurface microbial community and the biodegradation, right? Because again, it's another situation where biodegradation occurs from organisms that are out there in nature already. We're not seeding things. And the particular aspects of the environment can influence so much the speed of degradation and the efficiency of degradation and what products are left behind, exactly as you said a minute or two ago.
Yes, that's true. In fact, when we're looking at some of the organisms we're seeing come out of these wells, we're seeing some of the similar players that were cited in some of the Deepwater Horizon studies. Now, those environments are quite a bit different because one's relatively warm and the other's relatively cold, but they're both salt environments and they're both, deep water. And so you would expect there to be relatedness between some of these organisms, and in fact there is.
Oh, that's interesting. So give us an example of some of the organisms that are shared.
Oh, I knew you were going to ask that. Well, I can tell you a little bit more about the processes. So the oil that was released, you're going to have a complex array of different aliphatics and aromatics, and those are going to be degraded in different steps by different organismal groups. And in the shale environment, we have short hydrocarbons and also longer, more complex hydrocarbons, in addition to the compounds that are being sent down with the fracking fluids. And some of those constituents include petroleum distillates as well. And so those organisms are involved in the degradation of these different labeled complex carbon sources and in both environments.
Oh, that's cool. We have another question from the audience.
This is a question for Paula Moser. You mentioned how you took waters, did you mention soil bacteria and then feeding them with synthetic compounds that are probably part of fracking fluid, and the rate at which those compounds are being metabolized by that system under various conditions. I was wondering if you could comment on how the community of that, whatever you're studying, responded and shifted in response to whatever it is you were feeding it.
Absolutely. So what we're seeing is a shift in our great friends, the proteobacteria, delta, gamma, beta. And we also see an increase in some of the fermenters. So Clostridia and Bacilli are involved, especially in the first stages. So there is an immediate shift to be able to accommodate the degradation of these products. And that would be what we would expect from a lot of the release of hydrocarbons in the studies that have been done in gasoline spills and other oil spills in the near subsurface.
Does it revert?
Pardon?
Does it come back to the original composition anytime?
Boy, that's a great question. We haven't actually looked that long. That would be something that we should look into.
Thank you.
I think that really ties into thinking about this question. Why are these microbes that can degrade these hydrocarbons, why are they there in nature to begin with?
Well, they're there because there's all kinds of organic compounds that are coming from plants and animals that have these similar structures, and they've also been exposed to these hydrocarbons probably for a very long time through the evolutionary process. So they probably have a lot of these genes already, many of them. So once they see different chemical compositions and structures, it's probably easy for them to adjust and start to attack in a different way. What do you think, Erin?
Yes,
Would you agree?
Yes, I would agree with that. I think that's an excellent answer.
But it's particularly interesting to think that the organisms out in the ocean and the organisms in the subsurface really share these kinds of pretty complex metabolic pathways to degrade some of these compounds. It says a lot about the distribution of microbes on the Earth.
That's true.
Well, thank you very much, Paula and Erin, for joining us today. I think that a really important point of the conversation today is that microbes impact our lives in ways that we don't normally even think about. And an understanding of microbes in the environment can help us produce things that we want more effectively. Yep. It can help ensure the health of the environment, and ensuring the health of the environment is critical if we're going to have human health as well. Thank you very much.
Thank
Thanks
you.
to all of the visitors here and online. Please join us again for another version of ASM Live.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.